All right, thank you everybody for tuning in today. Again, happy Friday. This week's seminar speaker is Dr. Rachel Moore, who is currently a postdoctoral fellow here at Georgia Tech, working with Chris Carr. Dr. Moore is a microbiologist and astrobiologist whose research has focused on studying microbial aerosols and processes in different environments, including wildfires and rainfall. And I think we're going to be hearing a little bit more about that today. Although I do want to mention that more recently, while at Georgia Tech, she's been doing genome scale modeling to assess habitability on early Mars lakes as well. And we were talking about that offline before we got started here. And so doing more work on habitability, again, and how it pertains to early Mars. She received her bachelor's degree in molecular and cellular biology from the University of Illinois at Urbana-Champaign, and then a PhD in microbiology and cell science at the University of Florida in 2020. And so she did finish her PhD right in the middle of the pandemic, and then moved up here to Georgia Tech for her current position, I think in the fall of 2020. And again, where she's been working with Dr. Chris Carr. And so I'm excited that you're here to be sharing some of your PhD research with us. And thank you again for talking with us.
Yeah, thank you so much for that great intro. So today, like Dr. Francis said, I'm going to be talking to you about some work that I primarily did during my PhD program at the University of Florida. So I'm going to be talking to you about microbes and fire, and hopefully how this connects to astrobiology in that we are challenging the known limits of survival. This background image is actually a bunch of smoke that I was collecting in a fire in North Florida, I think in 2018 or 2019, and you can see just all these very large particles flying through the air while we were downwind of this burn. So just to start with a little bit of connection to astrobiology, since that is the topic of this seminar. One of the primary objectives of astrobiology, of course, is to search for life on planets beyond Earth, and often the search for signs of life involves studying the habitability of planets and moons like Mars, Venus, Europa, Enceladus, etc. And to detect the presence of life in outer space, we need to first define the limits of survival of life as we know it, because space has extremely harsh conditions like extremes in radiation, desiccation, temperatures, salinity, nutrients, and these conditions may seem deleterious for growth of life in outer space. However, microbes, they can be found inhabiting some of the seemingly inhospitable environments on Earth, like arid deserts, for example, the Atacama in Chile, hot springs, deep sea hydrothermal vents, lakes underneath lots and lots of ice in Antarctica, for example. So there's a lot of different analog environments to these extreme environments here on Earth. Some of those conditions, namely extremes in temperature and desiccation, are particularly relevant to the fire projects that I'll be talking to you about today. So in terms of astrobiology, there are several possible environments where those conditions may be particularly important to consider. For example, when thinking about Venus as a potentially habitable planet, as shown in this plot. This is just showing the surface of Venus and the temperature, how that changes with altitude. There are basically these layers of ever-present haze and stagnant clouds that have been hypothesized to be suitable for microbial life to persist in. A potential life cycle here would involve ranges in temperature depending on the mixing between these clouds and this haze, from temperate to quite hot, maybe even very cold, and also very, very low water content. Another example of an environment with possibly extreme desiccation and high temperature could be the subsurface of Mars. So this is a complicated phase model of the potential Martian biosphere, but basically all it's showing is depth below the surface of Mars and how that changes with temperature. So as you go deeper, it might be getting hotter, and also as you go deeper, it will be getting higher pressure as well. And what you're seeing here is these different phase regions are depicted. So the Mars phase region is depicted in this red hatched here. And then life on Earth is shown in green, so where that is found. And then also water is shown in blue, and Earth is matched up here with the brown. So you can see there's an extensive overlap between Mars and the inhabited regions of Earth right here. So based on these overlaps, the Mars subsurface and habitable Earth environments, or habitable subsurface environments on Earth there could be regions in the subsurface of Mars that could be habitable presently. And these might be hot and dry or hot and wet, depending on if water is available there. Also, the authors assume a temperature of 122 degrees Celsius, which I think is right here, is the maximum temperature compatible with life to survive, which is probably reasonable. I think this is based on deep sea hydrothermal vents, but is that the actual temperature, the actual maximum temperature at which life could survive and persist. So now I'm going to switch over to just a little bit of background on the Earth's atmosphere because it's important to understand more about this and the particles present there for understanding my projects today. Our atmosphere is actually considered an extreme environment for microbial life as we know it because microbes there are exposed to changes in temperature, pressure, radiation, and potentially low moisture and oxygen depending on the altitude. Typically, the higher up you go, the lower that's going to get, and you're going to have these differences in temperature as well. On average, we can typically find about 10,000 to 100,000 bacterial cells per cubic meter of air in the troposphere, and that is dependent on source. And of course, the troposphere is the layer of the atmosphere that we are in, and that's where we find all of our weather. There's also really no good way for cells to easily get out of the atmosphere unless they are rained out, so this is called wet deposition, or if they're attached to larger, heavier particles. They're simply too small to really be affected by gravity, and they can persist in the atmosphere for days to weeks or even longer if they get up into the stratosphere. So it's a really extreme environment and they are exposed to all these different stressors as well. Now I just want to point out before I talk any more about biological aerosols that total aerosols, they are typically measured in mass concentration in one of two subdivisions, PM10 and PM2.5, and PM just stands for particulate matter. 10 and 2.5 stands for 10 and 2.5 microns. So these encompass everything 10 microns in diameter and below and 2.5 microns in diameter and below. And these are also known as the coarse and fine fractions. Aerosols are typically monitored in this way as PM10 is inhalable, so you can get this into your mouth and into your lungs, and PM2.5 can actually penetrate into the gas exchange regions of your lungs, so this can affect humans through asthma, possibly lung cancer and other respiratory diseases. And bioaerosols like bacteria, for example, they typically fall within the PM2.5 range. Of course, we can have them fall into bigger ranges as well if they're attached to larger particles, or if we're thinking about maybe pollen grains, for example. Bioaerosols really encompasses everything that is in the air that's biological in origin, but typically we see them in the 2.5 range, and these of course can affect human health as well through if they're pathogenic, if they're allergenic, and also through the production of endotoxins. Another reason why aerosols are so important in the atmosphere, apart from health reasons, is because they are often required for ice crystal formation. And ice crystals are actually necessary for precipitation formation in temperate regions. For instance, supercooled water droplets that at sub-zero temperatures, warmer than minus 36 degrees Celsius, they actually do not freeze on their own. So the drop actually needs a particle to freeze at temperatures above minus 36 degrees Celsius. So for example, mineral dust, so this is what I'm showing here, a mineral particle or mineral dust in the atmosphere can actually serve as this particle to nucleate ice formation as warm as minus 15 degrees Celsius. And these particles facilitate the formation of ice crystals by serving as just a surface onto which water molecules can aggregate to form this structure. Now, certain bacteria, for example, like the Gamma Proteobacterium Pseudomonas syringae, which is a pretty well-known agricultural pathogen, it has specialized proteins that can facilitate the nucleation of ice at very warm temperatures, close to minus 2 degrees Celsius. So it's a lot warmer than most mineral dusts. So they do a better job at nucleating ice than those other dusts and also other biological particles like pollen and fungal spores. So both of these would still be referred to as ice nucleating particles because they are particles that can nucleate ice, but we can more specifically refer to the one on the bottom as a biological ice nucleating particle because the freezing was most likely caused by a protein. Now I mentioned that these atmospheric ice crystals are important because they are required for the production of snow and most rainfall. This occurs through the Bergeron process. Basically these ice crystals can grow through water vapor deposition, and once they become heavy enough they can fall out of the cloud as precipitation, so either as snow or they can melt into rain. Now here's finally where we start talking about fire. Fire is an important emission source of aerosols in general. In the US alone last year, I believe there were tens of thousands of fires, both prescribed, meaning that they were set with a purpose, an ecological reason, and also wildland fires, which are typically also set by people on accident. Together, they burned over 5 million acres or 20,000 square kilometers of land. For example, this is a map that I just generated from data from the Visible Infrared Imaging Radiometer Suite, which is on board the Suomi satellite. This is just showing fires that were detected since last November. So each red dot, I know it's a little bit hard to see because this data is so fine, but each dot is showing a 375 square meter pixel where an active fire was detected. So there are many fires across the U.S. producing aerosols all the time. And typically, since this has only been since last November, if I generated this map using data from the summertime, we'd probably see even more. Now, globally, each year there's about 3 million square kilometers of land that's burned, and wildfires in particular, sorry, that scared me. Wildfires in particular are responsible for the emissions of approximately 1,500 gigagrams of particulate matter. So huge, huge emission source, and it's a very important thing to study. Now, with the heat and particles emitted by the fire, sometimes clouds are formed that can produce precipitation. Briefly, fire can create a plume of turbulent air and smoke, and cooler air can then mix with that smoke as the plume rises, and then that can expand to form a cloud, which we will call a pyrocumulonimbus, for example, if it produces rain. So previous studies have actually shown that particles emitted during fire are active as ice-nucleating particles, and others have even seen freezing from smoke. Particles occur at warm temperatures, close to minus 12 degrees Celsius. Again, remember, warmer than what most mineral dust can freeze at. So we had reason to believe, just by looking at that data, that some part of those particles emitted were maybe biological in origin or microbial in origin. So we asked the question, is anything alive being emitted during wildfire, or something that was maybe once alive being emitted during wildfire? This would be, of course, an emission source for bacteria to enter the extreme environment of the atmosphere, but also it's a rather extreme mechanism of emission by itself. Fire, of course, is very hot and desiccating, and in this way, it may provide new insights for astrobiology in terms of the limits of survival. So to answer this question, is anything alive in smoke, we first embarked on a 2-year process of sampling air during prescribed fire, which again, you may have heard of this as controlled fire. So this is set for, again, an ecological purpose. And this was done at the Ordway-Swisher Biological Station, which is a 10,000 acre plot of land in North Florida. So this is a station that is basically used as a teaching grounds for maintaining natural communities. So this is a map of Ordway-Swisher, and I've just color-coded it to show the natural communities available across the station. So we have all these different types of forests and open plains and things like that. And the labels here are showing different units that I sampled at over the course of this study. This station is divided up into units, again, just for maintenance purposes, for teaching purposes. And so all these labels I collected at 8 different burns, and they were rather small fires. I mean, about 40 to maybe 100 acres at a time, which is pretty small if you consider those fires that you can see on the news, you know, where it's hundreds of acres at a time. And each of these fires were conducted at sandhill sites, which look pretty much like this over the 2017, 2018, and 2019 burn seasons. So this is what a sandhill site looks like. This is very typical of this part of Florida, where you see these really tall longleaf pine trees and a very open, nice understory that is maintained by this burning process. So this ecosystem has evolved with fire. So to collect aerosols, I sampled before and during the burn, so I could compare regular ambient air to smoke, right? And so to do this, I used volumetric air samplers to collect aerosols in 30-minute intervals. So this allowed me to collect about a half cubic meter of air on a filter like this or in a microcentrifuge tube. And you can really see just how many aerosols I was able to collect on this filter, because this starts out as white and it becomes fully saturated. And you can see just how smoky this is in the background as well. This sampler also collects PM10 data, which I was able to use to assess air quality. And then with these samples, I stained for total cell counts and viability. I cultured to count C-fuse and to identify just a few of the isolates. And then I did these freezing assays, which I can go into more detail if you're interested. But basically, these assays allow us to determine the concentrations of isonucleating particles, biological and total. And I also collected major vegetation types at a site. So I collected leaf litter and dry grasses. I collected green runner oak and saw palmetto. And our collaborator was able to then burn the combustion lab, which is basically like a big fume hood that you can burn stuff in. And then with those samples, I also I did cell counts and isonucleation assays, again, to determine the content of isonucleating particles. So finally, onto the data, starting with bioaerosol concentrations. So here I'm showing cells per cubic meter of air for ambient conditions. This is sampled, again, directly before the burn, and then during the burn, collected in the smoke. So these smoke-air mixtures, we saw that they had 5-fold higher cell concentrations than ambient air sampled directly before the burn. So this is a small difference, but it's a significant difference. There was about 60,000 cells on average per cubic meter of air in the smoke versus only 10,000 in ambient air. We also observed a significant 5-fold increase in biological IMP concentrations. Again, these are these isonucleating particles that are biological in origin. So we found about 2,000 bioIMPs in smoke per cubic meter of air versus about 500 bioIMPs per cubic meter of air in our ambient samples. And this is active at minus 15 degrees Celsius. Now, this number of biological IMPs in smoke made up about 80% of the total IMPs, so they were mostly biological in origin out of the total IMPs, and that was 2 times more than the percentage found in ambient air. So we found only about 43% of the total IMPs were found to be biological in ambient air. So this data combined indicated to us that fires were likely a point source of bioaerosols. Also, when cell abundance was adjusted to the PM10 data, again, this is the coarse particulate matter, we found that there were statistically larger numbers of cells per microgram of PM10 in the smoke-air mixtures as compared to ambient air. And this indicated to us that cells in smoke were likely attached to larger particles that were beyond the PM10 range. And we actually have microscopic evidence to support this. So here are some images that I took of our stained cells. So I used CyberGold, which stains things that contain DNA. So these are all images of cells that I collected during the burns, and then this is like a typical ambient cell image. And what you're seeing here is this bright diffuse green is some particle or matrix, perhaps a biofilm. We don't know the origin of this, but it's staining green, meaning there's probably extracellular DNA. All of these bright pinpoints that are circular and or rod-shaped and shiny, those are actually intact microbial cells. So you can see the cells that we sampled, they were often attached to these rather large particles in burns, beyond even 50 microns in diameter. And again, we don't know the origin of these particles, but this does indicate a difference in emissions because these particles are well beyond the PM10 range. Here are images depicting the viability stain that we use, CTC, which stains cells red if they have an active transport chain, meaning that they are active and viable. We found that 80% of the cells that we collected in ambient air and smoke were found to be viable by the stain. Of course, that means that there were significantly more viable cells in smoke than in ambient air, just because there were more cells in total. So this, again, was kind of an unexpected result for us. So this is where we start to challenge really the limits of survival, talking about viability. So survival for organismal life as we know it. Can microbes really survive and be aerosolized when the thing that they are living on or resting on is burning or about to burn? What temperatures are microbes actually subjected to? So this is an image from our collaborator, Dr. Lita Kobziar, who's a professor at the University of Idaho. And this is an image of a burn crew member standing next to this burning log. And this is that same scene, but viewed through a thermal camera. So you can actually see all of the temperatures associated with the actual burning log and around the zone of combustion. So you can see here that temperatures in this fire far exceed 122 degrees Celsius. You know, we can get up to 600 degrees Celsius. I think in more intense fires, you can even get up to 1,000 degrees Celsius. However, there is a high variability of temperature at sub-meter scales within and around this zone of combustion, as you can see here in this image. So at scales that are applicable to tiny particulates, some like microorganisms, they might actually be protected from super high exposure to heat and water loss just by the materials that they are attached to. Other cells, particles of mineral soil, plant tissue, maybe biofilms, for example. In addition, it's possible that the microbes found in smoke may be also advected from soils and plant surfaces from outside the combustion zone. So they might be basically advected in and mixed with the incompletely combusted particulate matter in the smoke plume as a result of pyroconvection. We just don't know the answer to that yet. However, we did test this in the lab, in that combustion lab. So if you remember, I collected individual fuels at a site that we burned them, in the laboratory setting. And I personally think that this points more to the microbes being emitted during the actual process of incomplete combustion, which is, you've seen this all happen if you sat around a campfire where there's a lot of smoke being produced, carbon monoxide being produced, that's incomplete combustion. I think that our results from the laboratory burns point more towards that being the actual process of emission as opposed to them solely being derived from the perimeter through convection. But again, we just don't know. But anyway, here are those combustion lab results. So we want to really investigate the burns of these specific biomass fuels to determine if fresh or decaying fuels produce more bioaerosols when burned. We had an inkling that probably decaying fuels would produce more because there are more cells per gram of soil or decaying biomass versus gram of fresh leaf. So here I'm showing you cells per microgram again of PM10 sampled during the burn. These three fuels, so grasses, soil over horizon, runner oak, saw palmetto, and we found that the laboratory experiments, they showed that the smoke from the combustion of the decaying fuels had emitted twofold more cells per microgram of PM10 compared to both of the fresh fuel types. And that same pattern was true, this pattern of emissions for CFUs that we collected and also IMPs, both biological and total isonucleating particles, although I'm just not showing that data here, but we found that decaying fuels did appear to significantly emit more bioaerosols per microgram of PM10 emitted. So with all of our field data collected from these North Florida sites, we were able to predict the magnitude of bioaerosol emissions from each of the 8 fires. So I'm going to just be showing you the average of those. On average, we predicted that about 16 grams of PM10 is emitted per square meter of burn, and we used that information and our cell and IMP data per microgram of PM10 that I showed you, and predicted that on average, 7 times 10 to the 9th cells and 10 to the 8th biological IMPs would be aerosolized per square meter of burn. So to put that into context, that's about 10 to the 15th cells and 10 to the 13th IMPs emitted per Ordway Swisher, that's OSBS, Ordway Swisher biological station burn on average. So these small fires, they can potentially mobilize the number of microbes that would be found in a metric ton of soil, which is huge. This is a huge emission source. But remember, we only investigated sandhill sites for this study, and these were also low intensity burns. We also always tried to sample in the smoke, but we were downwind from the actual fire for safety reasons. That was just what we were able to do in this case. So we wondered, does this happen for other types of fires, particularly for more intense fires in other regions? And also, is this just a near surface phenomenon? So specifically, do these microbes survive long enough to actually make it above the forest canopy, where they could be transported to even greater distances downwind? Perhaps they could colonize other areas. And if they can do this, what portion remains viable above the forest canopy? So this brings me to the next study that I'm going to talk about, which is more recent, where we sampled smoke during fires of much greater intensity, as you can see here. So these are actually also prescribed fires. They were conducted by the USDA Forest Service personnel in the Fish Lake National Forest in Utah. This happened between June 18th and June 20th of 2019. And the prescribed fire objective here was to regenerate aspen forest stands post-burn. So there was a lot of standing dead trees and down dead trees that were insect killed. So the vegetation here was largely alpine fir, subalpine fir, standing insect kill spruce, and patches of quaking aspen. And this area had not been burned for over 50 years. There was an outbreak of western spruce budworm, and both of these conditions had created a large down fuels, a buildup of these large down fuels, which contributed to this big burn intensity. So to actually ignite the fuel, the objective was to create crown fires, which means burning the tops of these trees, as you can see in these images. Crews actually used helicopter aerial ignitions, so basically shooting fire from a helicopter above the trees, and they also used ground-based torches as well. And this, I'm sure you can just see this already, but this is considered a high-intensity wildfire because the crown fire flame lengths range anywhere from 5 to 50 meters, and this is much bigger compared to the North Florida fires that were maybe 1 meter to 3 meters, I think, on average, so much bigger flames. And to collect air and smoke samples, my collaborators here designed unmanned aircraft systems, which were basically large drones that were fitted to hold vacuum pumps attached to button aerosol samplers that held polycarbonate filters, which is the same exact filter that I showed you previously, but this allowed them to basically get up into the smoke columns and travel with the smoke columns and always be directly above the fire, which was amazing in this case when it's a little bit too dangerous to get really close. And with these samples, I was able to then measure cell count, viability, and isonucleating particles using the same technique as before. We also extracted DNA and had it sequenced via Illumina-MiSeq to determine what was present in the smoke versus in ambient air in terms of bacteria, archaea, and eukarya, like fungi. So this is actually the first figure in our paper that just came out last week in ISME-Com. So this down here is the title of the paper if you're interested to see more about this, but this is just showing scaled concentrations of cells in ambient air and in smoke plumes. So we got 17 samples, I believe, of from the smoke, and 8 samples of ambient air sampled directly before the burn again. And we observed that the cell concentration in smoke sampled during those flights was significantly higher, again, than in air sampled prior to the burning. So I think it was about an order of magnitude difference. So about, I believe, 10 to the 5th versus 10 to the 4th cells. So pretty big difference. I also want to note that the concentrations of cells shown here is based on the total number of cells per volume of air sampled minus the number of cells observed in our procedural blanks. And also the number of viable and non-viable cells differed significantly between air types, again, because there were more cells in smoke versus ambient air. Both air types had relatively similar percentages, again, of viable cells. We found about 69, I think, to 78% viability, and this was just based on a live dead stain. So far, this is pretty similar to the data that we saw at Ordway-Swisher and those low-intensity burns in Florida. And I think I think the difference in cell count was just an order of magnitude higher, but we did go beyond that, beyond just counting for this study. We also did DNA extraction, and I'm showing you this here. So this is a really cool figure. It's really big from our paper again. I'm just going to start with explaining panel A. So this is the taxa composition from our analysis of our amplicon data from ambient air and smoke for bacteria, archaea, eukarya, and fungi. So bacteria in both smoke and ambient air we found was dominated by members of actinobacteria, which comprised about 50% and 36% respectively of those total sequences. We found archaea was only recovered in smoke, with Tom archaeota representing 4% or 0.4% of those of the reed abundance. And then we found Bacteroidetes, Chloroflexi, Planktomycetes, Acidobacteria. Those were all more abundant in smoke than in ambient air. Now I want to talk a bit more about actinobacteria, just because we found that in both ambient air and smoke samples, but actinobacteria, they were significantly enriched in smoke above ambient levels. For instance, certain actinobacteria families and alpha and beta proteobacteria families enriched in the smoke, they're often known to be associated with soils and the phylosphere. So this is consistent with these microbes originating from soil and local plants, which makes sense. I mean, if we're sampling smoke that's originating from soil and plants, it would also make sense, though, to see this in ambient air, because that is, again, probably the source for cells in this region. What I also found interesting is that we expected to see maybe an enrichment of endospore-forming bacteria in smoke, as that dormant tough structure could maybe survive temperatures and desiccation better, but that wasn't the case. We didn't see anything like that. And then in ambient air, we saw that Firmicutes was significantly higher, with Lactobacillus and Streptococcus found in higher reed abundance over smoke samples. Now, lactobacillus is a common fermentation bacteria or associated with fermentation. You may think of this when you eat your yogurt, but along with many streptococci, it's also found associated with oral microbiota. However, in this region, Lactobacillus and Streptococcus genera have been identified in environmental sources in the soil and amongst the plants, specifically for this part of Utah. So it makes sense to us that this could also be just found in ambient air. Now, if we focus on the chloroplastida, which I think is blue here in the eukarya from the 18S results, this is the mostly made up of the Pinnalis order, which is pine trees. It comprised the highest percentage of chloroplastida there. This suggests that, as would be expected, smoke from forest fires particularly partially comprised of plant materials, which is pretty neat to actually see pine DNA in the smoke. So with this study, this is the first time that genetic material from specific fuel sources has actually been shown to remain preserved in smoke, enough to actually uncover the source, which could be interesting for scientists that are conducting source tracking of smoke. So these Venn diagrams, they show total, unique, and shared phylotypes between ambient air and smoke samples for each sequencing region. Again, 16S for bacteria and archaea, and then 18S for eukarya and ITS for fungi. A phylotype is used in microbiology to mean groups of DNA sequences based on similarity. So each phylotype is representing a group of closely related sequences that could be the same or different species, but closely related again. And we can see that a higher proportion of bacteria phylotypes were unique to smoke than to ambient air. Only a small fraction was shared between both air types. And to me, that was somewhat surprising, as we thought that the ambient air would be much more similar in terms of sharing more of these unique phylotypes, ambient air is technically the background of the smoke and air mixtures, right? We're just adding smoke to this ambient air. So we thought that there would be more similarity there because there would be enough mixing. So that's the case for the bacteria samples here, and we also see the same trend for ITS, where there's more unique to smoke. ITS here, more unique to smoke, and 18S as well. Where there's only a small fraction shared between the ambient and smoke samples. So here in panel C are PCOA plots of community similarity or dissimilarity between ambient air and smoke. And in panel D, we have a diversity comparison. Panel D, we have a diversity comparison between ambient air and smoke, where error bars just represent standard air. So in these two panels, we can see that bacterial libraries differed significantly between ambient air and smoke in regards to community structure, shown here in C, and diversity, shown here in D. So for instance, you can see that the ambient and smoke bacterial libraries, they don't overlap much in the PCOA plots. And the smoke libraries are also much more diverse, you can see in the orange here, than ambient air, and this is significant. This again is also true for the 18S libraries, although at a weaker significance level, but it's not observed with fungi for the ITS libraries, and we think that the reason for this potentially comes down to just challenges with characterization. So 42% of the fungal families that we found in smoke and 39% of those in ambient air were unclassified. So this may simply be due to just limited reference libraries, but we don't really know. But seen all together with observations that these fires are aerosolizing viable microbes, the molecular results show that bacteria and archaea assemblages in smoke are more diverse and distinct from those found in ambient air, which I think is pretty exciting. This is the first time that we've been able to show this in a publication. So now we know fire is probably an emission source or is causing the emission of microbes, the majority of which remain viable. We've also seen that microbes, the assemblage of microbes in smoke, tends to be more diverse and distinct than in background air. So how do the burns in Utah, those high intensity burns, compare to the lower intensity burns of Florida? Remember in Florida that I estimated 10 to the 9 cells per square meter of burn were emitted, which is about 4 times 10 to the 13th cells per hectare of burn. Now in Utah, with these high intensity burns and greater amounts of dead fuels, we saw about an order of magnitude more cells emitted per cubic meter of air. So here I estimate that about 10 to the 14th cells were aerosolized per hectare of burn. So these burns in Utah, they covered about 4,200 hectares, which is a huge, huge burn. That's about the size of Ordway Swisher in total. We can estimate from this that they mobilize an estimated 10 to the 17th cells, or 100 quadrillion cells in total. Again, this is just a rough estimate, but that is a huge amount of cells, especially if a significant number can remain viable. But again, one thing to keep in mind is that we don't actually know yet how these microbes are being aerosolized within and or near the combustion zone, just that this emission tends to significantly occur when fire is happening. So this will take further research. But this research, which we are calling now the field of pyroaerobiology, I think it could reveal new insights about the tolerance of specific organisms to the environmental stressors associated with the combustion environment, like extreme heat, desiccation, and even aerosolization. And today I talked to you about these two studies highlighted in yellow. So the prescribed fires in northern Florida, and then also that burn in Utah. But there are many other studies currently being conducted by my collaborators, and I believe they have papers currently in prep, which is very exciting. For example, the Kansas tallgrass prairie, the Kanza prairie. This is going to be a really cool project because it's a different type of burn, being that it's not in a forest. This is a prairie region. And with this study, they're also including smoke treatments of sterile soil to see if microbes can actually colonize new locations downstream, to see if this actually has an impact in other locations. It actually helped to write a small part of this NSF grant proposal. And after I joined the CAR lab here at Georgia Tech, I got funded, which is really exciting for them. So I'm really excited to see where they go with this. And that's being led by Professor Lita Kobziar and Brent Christner, who was my PhD advisor. So I'm really excited for this field to see where it goes. So with that, I would like to say thank you to everybody that has been part of these projects, all of the burn crews that I cannot list here because there are too many of them to name. And of course, funding through USDA, NIFA, NSF, and thank you all for listening as well. And any questions I will be happy to take.